Genome is genomes aggregation database, and it's the collection of the research article and related content from the genomes, and it used to help describe the analysis of the human genetic variations. And the gene we're gonna study in this tutorial is GATA6 genes. So you just search it up from browser, the homepage, and then we're gonna bring you to the page about these genes. And in the first, it gives you the ensemble gene IDs and all the ensemble informations, and also link to the external resource of ensemble and UCSC browsers. And on here, we'll be showing you the synonymous and missense and POF values. And also in here, they're gonna give you the overlay of the genomes. And the genome is overlapping with the exome. And you can see that the green is overlapped with the blue. And also, you can also download the safe plot. And in here is the soul transcript. And you can see that the coding part is the black, and the untranslated region is the white box will be, and this line just connecting them. And also in here, that's what's interesting about. So you can select the variations, the variants of the pathogens, likely pathogens, or like uncertain significance, or like benign, slightly blind. So you can link to it. And then, so if you are interested in studying pathogens, so you can just only check on this check mark and then leave the rest like this. So you can see that will be displayed like how many pathogens from this 30 study variant they had. Like, so that's the 30 they had. So you can see that that's one pathogen. So you can also see from expanding this. So you can see on this, they can also give information whether or not it's pathogenous and like type of the mutation it is, those friendships, and yeah. If you also click on the uncertain, they'll give you more, and also benign, likely benign. So the only thing we're interested in is the pathogens. So then you just look at this to see. And if you click on all, they're gonna show you all. And the bottom one is the variants. Look at more detailedly, like showing you what is the variant ID and the source can come from, and also the consequence of how you change, and the VEP annotations, the variant effect predictors, and the LOF variants, and the clinical significance, like likely benign or like uncertain significance, and the flag, and the real counts is like how many of them is like the real counts and the real numbers. So that will be a good source. And then you can use this browser to get the information. Also, you can download this, export the variants to CVS files. So that's how I got the files from. So that will be downloads. And also I can also try to the TP53 gene, so you can see like this. So you have to give the information about these genes, like how the mutations, like type of the mutation they had. So you can see that for this mutation, this gene, they have more mutation than the GATA6 gene, because that's more popular for mutation causing cancer. So the next, I'm gonna also show you that in the ensemble website that is showing you the information of the GATA6 genes, and like also this gene legal and all this. So if you want to get more information about the genes, so you can also use ensemble. And then the next, I'm going to talk about the package I'm using in the R. So it's G3 visualizations. So it's a package that of the art that can be visualized genetic mutation data using the lollipop diagrams. So the first part you're gonna do is like install this package, and then you're gonna roll the library, and then the first one is giving you example that if you're using the MAF files, so if you're using the MAF files, that's how you do it. And if you only had the CSV files or the TSV file, then you're gonna use this pathway, and the code is like different, very different than the MAF files. So when you generate the lollipops diagrams, that's how it look like. So you're gonna give you like how many counters, positions, and raise the others to the lollipop. The circle will be showing the how many count of the mutations occurring on this whole genome layout. And also another useful is like you can get the mutation information from the CBio portals. CBio portal is another website that you can get the cancer genomic data sets. So that's how you're gonna run it if you get it from the CBio portals browsers. It also had the similarity of the lollipop diagrams too. And yep, the rest just information, so you can get it if you just look up from the CRAM websites. Yeah, that's all I introduced about browsers and the package I use. Thank you.